hopefully a couple of people will still join us. May said we've got maybe around 24 people registered today. We've got a very, very exciting set of talks today, followed by hopefully will be a very fruitful discussion with some questions that both of our speakers have come up with. And so the topic today is on remote sensing and mixotrophy. And this is a topic that I think's come up a little bit in some discussions, just this idea of like, got all this remote sensing stuff. It's this, you know, there's a lot of rapid stuff happening and advancement in the field of remote sensing. Is there any future with helping us study mixotrophs through remote sensing, what that might look like? And so, you know, this might be one of the first few discussions of its kind here, and see what comes out of it. And who knows, maybe in 10 years we'll have our own export style project where we're studying mixotrophs from space. And it all starts right here. So let that, you know, inspire you today during the discussions. Our first speaker that we have is Dr. Jessie Turner, who is currently a postdoc at UConn with Heidi Dearson. And she's, at least what I've seen from her work there, is working a lot of stuff with remote sensing data down in the Western Antarctic Peninsula. But she recently got her PhD from here at VIMS, where I first met her. And we don't really have a remote sensing expert at VIMS at all, but she came in, wanted to do remote sensing, and did remote sensing anyway, despite there not being someone here for that. So she's taught herself a lot and went out of her way to become the expert that she is in remote sensing. So she's going to talk to us, kind of introducing us to just remote sensing, some of the information behind it, to get any non-remote sensing experts in the audience up to speed on that. So I'll turn it over to Jessie now.
Thanks, Nicole. Wow. It's nice to see everybody's little names popping up. And if I haven't met you yet, it's nice to e-meet you. Here's the screen. Right, so my name is Jessie. Nicole introduced me. I'm kind of Connecticut and VIMS both. And I like to start out with a picture of Chesapeake Bay, because that's where I did my most recent work. And look at all the colors. There's a lot of colors, right? There's the blue open ocean. There's the green of the coastal waters. There's sort of this yellowy brown up in the rivers from all the sediments. And then you can even see some brown sedum from the marshes, and this green phytoplankton, that sweet phytoplankton. So we've got a lot of colors, and satellites can help us see what's going on by way of those colors. So when I first started looking at satellite data, I thought, wow, this is great. Like, I was stuck with these cruise stations, which I loved my cruise stations, my cruise data, but spatially, it just wasn't quite enough. You know, I wanted to know what was going on through the whole Chesapeake Bay. And satellite data had that spatial coverage. So I thought, wow, this is just great. So that's sort of the really obvious advantage, right? Is the coverage. But then I started talking to people, and they were in two camps. One set of people would tell me, oh, how great, you should use satellite data. You know, it has this long time series, it's all available online, you're going to do so well. And then this other camp of people would say, you can't use satellite data, it's way too much error, it's too complex, there's too many clouds, it only gets the surface ocean, you're not going to be able to answer your question. Well, neither person was wrong, right? There's some truth in both of these sides. So I've landed on use it, but know the pros and cons of what it is that you're using. And so hopefully this will help you out with that. Has anybody ever been shopping in a foreign country? Like, you go to this foreign country and you have to grocery shop for the first time, and you just think, oh my gosh, I have no idea what I'm looking at. What does it mean? So that's how I felt the first time I went in to download satellite data many years ago. And maybe you felt this way. You don't know what it is. You don't know what the acronyms mean. You don't know what to choose. And so we're going to get into it a little bit to decipher that. So for satellites, you have two main types of satellites by their orbit. Polar orbiting, where the satellite goes around the Earth over the north and south poles. As the Earth rotates, the satellite keeps moving around the Earth like winding a ball of yarn, and most of them are polar orbiting that we'll use. They can either map a wide swath of Earth's surface like satellites with a daily overpass, which gives you low spatial but high temporal resolution, or they can map a narrower swath like satellites with a 2-week overpass, which give you high spatial resolution but low temporal. The other type of satellite is a geostationary satellite, where the satellite goes around the Earth at exactly the same speed as Earth rotates, and it looks at one spot on Earth. Whereas the polar orbiter looks at the whole Earth over some time interval, and the geostationary looks at one spot all the time. An example of one of the geostationary is GOES, stands for literally Geostationary Operational Environmental Satellite. An example of a wide swath polar orbiter, Aqua or Terra, and a narrow swath polar orbiter is Landsat. Next, we have the sensors. Sensors are also called instruments. On the same satellite, you might have many sensors. For example, an infrared sensor and a multispectral imaging spectroradiometer. Things like TIRS on Landsat stands for thermal infrared sensor, or MODIS on Aqua and Terra. So know that you can have the exact same sensor like MODIS on board multiple satellites like Aqua and Terra. Next, let's talk about data products, the ones we most commonly use for the ocean. First, there's remote sensing reflectance, or RRS. You can think about it as the brightness of the Earth's surface at specific colors, like the color red. It's the ratio of the light coming out of a surface to the light going into that surface. Or LU, the U is for upwelling, to ED, the D is for downwelling. Upwelling light coming out is sensed in just one direction, whereas ED is from all directions. So the units of RRS, when you look at it on a graph, represent that it's reflectance at one angle, hence steradians. And it's the brightness of each specific color or at specific wavelengths. For example, red band RRS is associated with water turbidity or water clarity. This was in some of the aquaculture papers. This is what I study in my research. So the more red it is, the more stuff is in the water. The water exhibits more brightness at that specific color. And here, the color red means wavelengths 600 to 700 nanometers or so. We could pick a wavelength measured by a satellite like 645 nanometers to look at water turbidity. And this is why it works. If you look at the full spectrum of remote sensing reflectance for two types of water, open ocean water, which is clear, versus Chesapeake Bay water, which is turbid, at that 645 nanometer wavelengths, the turbid water will have higher RRS or deliver more red light up to the satellite sensor. Next up, we have a crowd favorite, chlorophyll. People want to know how green is the water, because that's a proxy for phytoplankton pigments. And it can tell you how much primary production is going on. We get chlorophyll from the ratio of RRS at the green to RRS at the blue wavelengths. For example, if the satellite measures green at 550 nanometers
and blue at 412 nanometers. We then take the ratio of those two RRS values. Multiply them by some coefficients and use that to estimate chlorophyll concentration in milligrams of pigment per meter cubed of ocean water. Again, if we look at open ocean water versus Chesapeake Bay water, full spectrum, you can tell from where those blue and green values are that the bay is going to have higher chlorophyll.
Great, so maybe, I'll turn my volume down. Maybe you're thinking, oh, I already know that, but I think it's a nice review anyway. So if you ever want to use that for a class, it's on YouTube. I'm working on my YouTube channel here. So most of my talk is going to be passive remote sensing, and ocean color is passive remote sensing. And that just means it relies on light from the sun. Whereas active, like LiDAR or radar, you're beaming down a signal and then seeing what comes back to the sensor. And so I'm mostly talking about passive, but there's a couple things we might be able to use the LiDAR for, and I'll talk about that later. So again, reflectance, it's a measure of color. And as you go from the open ocean, it's very blue, it's very bright in the blue part of the spectrum. And then as you go to coastal water, it gets greener and darker as the phytoplankton and sea dom absorb that blue light. And then if you move way up into the estuaries, it gets kind of yellow to brown even. And that's because you have sediments and detritus that are scattering light and making the water look brighter. And satellites, typically, there are some exceptions, but typically they measure this color at certain bands or certain wavelengths. And this is MODIS, for example. So just 3 examples. These are maps of lower Chesapeake Bay. And if you go left to right, you're getting better spatial resolution. If you go right to left, you're getting better timing or more frequently passing over the same spot. So you could get daily coverage if there were no clouds. So there are trade-offs here, right? You can't have everything. And so your research question should determine what sensor you use. I could say that over and over again, because sometimes we forget. One example of how to do that. So this is by Dick Stump, and he wanted to look at phytoplankton blooms, and he wanted to look at the red and red edge part of the spectrum. And so he made a table of, okay, what sensors are available? That's on the left here. What's the sensor? And then he said, okay, let's rate them by their spatial, their temporal, and their spectral resolutions. What do we need? And so it's color-coded, green is good, red is bad. And according to this table, it's kind of a lot to digest, but this top row, this is a European Space Agency satellite that currently has 2 satellites, so you get a more frequent revisit. It's called OLCI, O-L-C-I. That was sort of the winner for him for this phytoplankton question, because it had 300-meter spatial resolution, which is kind of medium yellow, had a good temporal timing, every 2 days was good enough for what he was asking. And then it had these red edge bands that were really important for phytoplankton. And you might think, okay, what is the red edge? That was not in my whiteboard video. Well, here it is. So usually we talk about green to blue ratio for phytoplankton, and that's sort of like the historical, how green is the water, how much phytoplankton is there. But the red to red edge part of the spectrum is actually really helpful because that's where chlorophyll fluoresces, right? We know this from just doing benchtop fluorometry, right? So if you have a satellite that has 3 bands in a sort of triangle shape, and so the examples here are these 3 red dots, you can get this fluorescent line height is what it's called. So if you ever see the acronym FLH, that's fluorescent line height. And it's just a way to measure or estimate at least chlorophyll fluorescence from the satellite signal. So that's kind of powerful. And so this is one example, again, in Chesapeake Bay, of getting some halves from that fluorescence area of the spectrum. And so these are 3 different products that are all from that red edge portion. And the rightmost one, this is Shelley Tomlinson's work at NOAA. And she found from this sort of non-fluorescing product that's usually used for cyanobacteria, it picked out some hot spots on the map for various different species. And I'm not a mixotroph expert by any means, but I know a couple of those are mixotrophs, right? Woohoo, I got them in my talk. So Porocentrum, and we call this one Carl in Chesapeake Bay. Key point, you still do need an in situ sample to verify what species that is. We're not quite there yet, but it's a way to map, you know, some hot spots or where things might be popping up. So it's just one tool in the toolbox. Another thing I want to mention, and maybe you've heard of some of these things, but the future for satellite ocean color is really going to be targeting some phytoplankton research questions. And that's because these future sensors are hyperspectral. So the first one that's going up in just a couple of years here is PACE. It's plankton, aerosol, cloud, and ocean ecosystems. It's coarser on the spatial side, but it is hyperspectral and it's going to have daily overpass. And so that's going to be really powerful. And then Nicole mentioned maybe in 10 years, we'll have some kind of exports like thing. Well, if you're thinking 10 years out, I know I am, SBG, surface biology and geology, that's going to be finer spatial resolution. So they haven't really decided yet, I don't think. If someone can correct me if you know more, but maybe 100-meter spatial resolution. So that's really powerful if you work in coastal areas and you're wanting to look at these things. And I'm involved with the PACE group, so feel free to ask me questions about it. I'm helping teach a class in Baltimore in August, and we're gonna actually have a lecture where we go into, they have fake simulated PACE data, like what will come out of this satellite, it's simulated, and we're gonna have a little walkthrough of the data, and that's available. So if you're writing a proposal and you wanna use that, and you wanna watch this recorded lecture after August, shoot me an email and I'll tell you where to find it. Example, so hyperspectral. How is that important? Why do we care about that? This is one example. And look, guys, another mixotroph. I got another one. Mesodinium in Long Island Sound. It was a big red tide happened this summer, a few years ago, and they were able to look at that from this hyperspectral sensor, HICO, H-I-C-O, that used to be on the space station. And this is a weird species. It fluoresces bright yellow. And they were actually able to see that with HICO, which is hyperspectral, but they wouldn't have been able to see it with these heritage satellites that we use a lot, like Modus Aqua, Modus Terra, because they don't have a yellow band. And so this is where hyperspectral, nature has some stuff out there that you can't predict what you're going to be able to see unless you have the data. So we need to think really creatively with these future satellites. And then I have a couple more examples that are just sort of things I wanted to throw in here. I am not an expert on this stuff, but I wanted to direct you to these papers to kind of get your mind thinking about it. LiDAR, where you beam light down and see what signal comes back to the sensor. That could possibly be used to get at these diel questions. Again, I'm not an expert at this, but there's a sensor called Calliope that was on the satellite Calypso, and that was able to get this signal of day versus night phytoplankton, because it still gets a signal back even at night, unlike passive where we're relying on the sun. And so this could be something to look into. Another thing to look into is there's a possibility that we could get grazing rates from satellite. It's in its infancy still, but it's based on sort of this idea that particle size changes back and forth depending on who's grazing on who. And particle size is something we can estimate from satellites from the blue light backscatter. So again, I'm just kind of throwing these ideas out. Feel free to look up these papers and just to get our brains working. But in summary, you've got your advantages and disadvantages. And I always say at the end of any talk about remote sensing that it can complement, but never replace the in situ data. Like we're always going to need that microscope, no matter how fancy we get with our satellites. So with that, I'll take any questions. Thank you.
Thank you so much, Jessie. That was excellent overview. And thank you for the plug of microscopes at the end too, because I do love my microscope. We can have time for a couple quick questions if anyone would like to ask Jessie. I can get us started because you said you were you could answer questions about PACE. I was wondering if back to that table of the frequency of all those different things, where pace might fall on that.
Yes. So it would be if it could be even brighter green on the key spectral, it would be. It's like, wow, hyperspectral. As far as spatial, it is a red box for spatial. It's 1 kilometer. But it's green for temporal, so it's 1 to 2 day temporal. So yes.
Thank you.
Yeah.
All right, then. I guess we will move on to our next speaker. And so Dr. Sara Rivero-Calle is at the University of Georgia's Skidaway Institute of Oceanography down in Savannah. And we were talking beforehand, she started that position in August 2020. So like me, she's got to enjoy the perks of, being remote for essentially most of her time as an assistant professor. But she obviously does work with remote sensing and is very interested in application side of the remote sensing data with phytoplankton, and has at least as much of an interest as anyone can these days with mixotrophy and remote sensing. So I invited her to speak on that topic. So I will turn it over to her now.
Thank you, Nicole, for this introduction and for the opportunity to talk with you all. As I mentioned to you a couple of times, I'm very interested in mixotrophy and what we can do with satellite remote sensing. So I'll share my screen. Oh, no, not that. Sorry. Share PowerPoint. Okay. Okay, so thank you, Jessie, for a great intro. You'll see that some of the topics that she mentions and her beautiful descriptions will also appear here. But I'm very excited to share with you some of the ideas and advantages, disadvantages. One of the purposes of my talk is to remind you that we're looking at microscopic stuff and we're trying to see that from space, and B, demystify what we can and cannot do with remote sensing and give you the idea that there's just so much room for things to do. Okay, so basically, like I said, I'm going to talk a little bit about the obvious advantages, the technical limitations, some of the science behind remote sensing and algorithms, some approaches that have been taken in the past, mostly geared towards phytoplankton, some considerations about resolution, which is what we've been talking a little bit about, that trade-off, and then something I'm working on currently in terms of mixotrophs. So obviously, I don't think I need to say this again. We all know that these beautiful long-term, large-scale images can be provided with remote sensing. So that's a great advantage, but it doesn't come without limitations. And so I want you to be aware that if we are using a passive sensor that needs sunlight, you need to be aware that you can't get information at night or in the poles during the wintertime. It's just not possible. If you're using an active sensor, you might be able to get some data, but otherwise you cannot. The other thing is, if you're using a passive sensor and you have clouds, you're not going to get a signal. And I'm showing this image here that shows 1 day worth of data from MODIS. And while you can get data from around the world, you see that we get nothing in the Southern Ocean because this is during the wintertime in the Southern Ocean. You also have those missing data due to clouds, and you also see those streaks, so those lines, that has to do with the way the satellite orbits around the Earth. And so this is going to limit what we can and cannot do. But the other big thing that you need to take into account is the vertical dimension. So if you're interested in having detailed information at different depths, that's going to be really complicated because these type of satellites, they integrate over the water column for as the best light can penetrate in the water. So those are some of the things that you need to be aware. Remote sensing is powerful, but it has many limitations as well. And then I'm going to jump into how do we actually get these kind of images. This is an image of chlorophyll for the entire MODIS mission. So basically, remember that ocean color, which is a passive technique, what it does is it records the reflected sunlight from the surface of the ocean. And the different colors represent how light interacts with different materials. And with that, we get information about blooms, sediments, organic matter, and water quality. So how does this actually work? And so we have our sun, we've got sun shining down on the water, and two things can happen. It can be absorbed by the water itself or materials in the water or plankton, or it can be scattered around. And some of the scattering will go up, and that's what the satellite will actually collect. And so it's this balance of forces of what comes down, what goes up, what gets absorbed and scattered that we use to get information about what's in the water. But you also have to take into account that it's not just how much, but what the quality of the water, of the signal is. And so if you remember from your physics classes, the white light breaks down into all the different rainbow colors. And so those are the different wavelengths, and different wavelengths will penetrate into the water and react with what's in the water differently. And so we record those changes by different colors, different wavelengths to get at what's going on in the water. So these are some examples, again, similar to what Jessie showed of how there's different ocean colors and different spectra related to them. And that's what the satellite is actually seeing, its wavelength versus water leaving radiance. But also remember that as much as long as the, or as well as the absorption and scattering processes are happening in the water, there's also just as much happening in the atmosphere. So what we are actually retrieving with satellites is about 1% of what is emitted by the sun. So it is amazing how much we can do with just that 1%. And now I'm going to give you an example of a multispectral satellite, which I'm directly linked to. And so I'm going to do some shameless self-advertising. I'm part of a group that launched the first dedicated ocean color multispectral CubeSat, Seahawk, and its sensor, it's called Hawkeye. And so here's zooming into what the sensor looks like. And so there's 8 slots, and through each one of those, we're measuring a different wavelength. So in essence, we're measuring 8 wavelengths, 8 bands. And I've added color to each of them so that you have a sense of what those numbers mean. So 412 is blue and 865 would be red. And it's pretty much a copy of SeaWeave's, but our satellite is the size of a toaster. It's really this size, it's tiny. And yet we can get some really high quality data. So with each of these slits, we are recording the brightness at each one of those bands. And so it's basically a picture of the same scene, but looking at a different wavelength, and it's in a shade of gray. We can combine them into what is more easy for us to interpret as RGB, and we can get what we call a true color image. So this is a scene of Greece, for example, and we can look closer at one of those pixels in the blue. And again, you see wavelength versus reflectance or radiance, and you'll see that you have very bright signal in the blue, the water is very blue, and then low signal in the red. And if you look at a green pixel, then you'll have lower in the blue and bigger in the green. And so it's just that combination of blue to green that initially was used to calculate chlorophyll. It's a ratio between the two using empirical data. Again, we needed to go and take in situ data. But so that's as simple as that. You can build an algorithm and you can get a proxy for chlorophyll. Now I'm going to describe some of the approaches that we could take for mixotrophs based on what we've learned for phytoplankton groups. And so you can either use an electromagnetic signal, remember what we're talking about, absorption, scattering, and radiance, or we can look at a proxy or using chlorophyll as a proxy, or we can make predictions, we can build algorithms or models based on remotely sense-derived drivers and our prior knowledge. And I want to emphasize this prior knowledge because that is very important as to which sensor we pick, what kind of algorithm we build, what type of spatial resolution you're going to need. So I'm going to go over some of these approaches. And if you haven't read this review by Colleen Mao at all, I highly recommend it as the first intro on how you can use those different approaches to get at phytoplankton taxonomic classes or size distribution. And if you look at the third tier of boxes, remember that we talked about abundance-based or chlorophyll, radiance-based, absorption-based, or scattering-based. So these are all using some of these approaches, and you have some examples below. And now I'm going to turn to one of these iconic groups. I did my PhD on Caucalithophores, so this is near and dear to my heart. And I just want to explain once again why algorithms and satellite remote sensing is not witchcraft. It's basically another ratio of algorithms and in situ data that we've used to create some sort of relationship. And so there's the two-band approach to calculate PIC, which we use as a proxy for Caucalithophores, and the three-band. So one of them is using water leaving radiances in two bands, and the other one's using top of the atmosphere reflectances with three bands. And this is just an example, but NASA, for example, has published in these ATBD documents exactly how each standard algorithm for a standard product is being used. So you can reproduce those algorithms. Another example of how we use remote sensing for HABs is the typical Carenia brevis that we find in Florida, among other places. This is an image, I believe, from South Africa. And so you see that there's a clear ocean color change there, right? And in this case, I also wanted to show you a very simple algorithm where they were looking again at the band 3 and band 14 of MODIS. Those were some of the ones that Jessie was mentioning for the FLH. And so this difference between the two bands can tell us whether it's Carenia brevis or not. And it's used in a way where you just subtract one band from the other and divide it by the sum of the two. This is similar to the NDVI algorithm, if you've ever heard of that on land. And so just based on that, you can start creating maps. The uncertainty associated with it, or how good that product is up for discussion, but it gives us eyes for a large scale region now. So that's what I want you to understand. And then I want to show you another iconic plankton group, Trichodesmium. And it also changes the color of the water. But there's something that I want to discuss with this example. And one of them is detectability limit. So for example, out here in the South Atlantic bight, we see Trichodesmium, or we see trichomes all the time, but they are not in a high enough concentration for us to see it from space. So that's something that you need to be aware of, and that's where the spatial resolution also comes in. The other thing is that you need to have a signal at the specific bands that we need for that particular organism. So in the case of Trichodesmium, the key thing about them is their phycobilic pigments. And so you need to have bands in the 550 to 650 nanometer region. So Makina in 2015, for example, he characterized the absorption and the backscattering multispectrally of these two organisms and of this organism. And he then looked at what the available sensors, ocean color sensors, were at the time and whether they had the right bands for what he needed to study. So this is how I want you to think about how you can use this for your particular Mixotroph study. The other approach that we mentioned is using chlorophyll and using chlorophyll concentration, a function of it, to describe phytoplankton taxonomic and size classes. And so this is some work from Hirata et al. Where he was able to do both. And again, it's basically using in situ data, empirical data, to build this formula that are all a function of chlorophyll with different coefficients. And then lastly, I want to give you the idea as well that we can use remote sensing derived drivers to get at a model to predict where it could be, where we might not have an electromagnetic signal coming out, but we can use other environmental drivers and background knowledge to get at having some sort of a map, a global or large regional map of where we could find a mixotroph, or for example, I'm just throwing out there a percentage of mixotrophy. And as we said, we're very excited about new upcoming hyperspectral missions. We had a previous hyperspectral sensor on the International Space Flight Station, HICO, a few years ago. But that was a sensor on the International Space Station. We're now going to launch in the next, it says here 2023, but I think it's more 2024. We'll be launching PACE, which has hyperspectral 1 kilometer resolution, 1 2-day repeat. So what we were seeing with those missing bands for that particular organism, it's going to change the way we think about things. And the other upcoming mission is Glimmer, which will also be 1 kilometer resolution, and hyperspectral, but in this case, it's going to be geostationary, and it's going to look at the Southeast US. So I'm very excited about that one, because this one is going to give me repeat measurements every, I don't know if it's every hour or 2 hours. So we can start looking at rates. But again, we're limited by the regional aspect. And if it's not clear enough from what we've both said, we're very excited about getting hyperspectral data, because our spectrum or our spectra are going to change significantly. And that could make the difference between not able to distinguish HABs from non-HABs to actually being able to distinguish them. And so this could also give us an extra edge for our mixotrophy interests. One other thing that my group is working on is actually characterizing optical properties of HABs. And in particular, we're working with a prototype that is able to capture hyperspectral backscattering in lab settings, which is a completely new thing. So we're hoping that we'll be able to get more of that optical information that is not just related to our pigments, but also to shape and size. So this is work in progress. Stay tuned. But I want to bring you back to the idea of the CubeSat that I was mentioning, this miniaturized ocean color sensor. And we just got funded for 3 years of using the Seahawk data to demonstrate its capabilities. And so I thought, why not look at a Mixotroph to do so? And so I am currently starting on this project to look at Noctiluca and exploiting some unique characteristics of Seahawk to get at a better understanding of Noctiluca. And so it comes down to what we were saying before. You have that trade-off between your spatial scale, temporal scale, and spectral resolution scale. And CubeSats in particular are in this weird position where they can, they have the capability of getting high spatial resolution. But if you don't have a spatial and temporal resolution, if you have a constellation, but if you don't, it gets tricky because, for example, in our case, our repeat time is every 3 weeks. However, there's tricks to that, and I'll show you in a minute. If you want to learn more about the Seahawk CubeSat mission, I've added there the link. But the main thing is that if we compare MODIS to Seahawk, remember that MODIS is the size of a bus, and it was many millions to develop it, many years to launch it and get it running. And it's awesome. And we have now 2 decades worth of data, but we're working with 1 kilometer pixels. The advantages of the CubeSat is that we're now looking at an order of magnitude higher spatial resolution and just as good quality and at much reduced cost. So we're trying to demonstrate that with CubeSats, we could fill in the gaps in between larger missions. And I'm interested in demonstrating that, as well as the capabilities that can be acquired with a satellite that we can actually tell to tilt. So instead of having a repeat time or revisit time of 3 weeks, we can now get daily images. And so this is an experiment where we looked at a whole week in the Arabian Sea looking for Noctiluca. Of course, here you can also see around midweek where we also have a dust storm, so that did not help us much, but I'm trying to demonstrate how useful these low-cost CubeSats can be for remote sensing. One of the things that really interested me about using Noctiluca is because I always think about changes in ocean color. We've got a Noctiluca that can appear green or can appear red, and it has endosymbionts in there. And to make things even more interesting, it also bioluminescence at night. So I'm very interested in characterizing the optical properties of Noctiluca, and we don't have it in culture right now. So if any of you have them in your lab, please contact me. I'd love to use it for a new prototype instrument and create some spectral libraries of it. But my main question is not so much about the Mixotroph itself, but to demonstrate the technology. And so I was interested in looking at Noctiluca in this area, because if we compare what we can obtain with Hawkeye versus MODIS, you can see that that added order of magnitude spatial resolution can make a huge difference, but we don't know by how much. We want to quantify that information gain and understand better the subpixel variability. And so I'm going to be comparing what we're seeing with Hawkeye with other instruments with higher or different spatial and temporal resolutions and see what we can learn from that. But in general, when you're looking at, okay, I want to study a Mixotroph, like Jessie was saying, you have to sit down and do your table and see, okay, how much temporal resolution do I need? How much spatial resolution do I need? And what is the spectral resolution that I need? And so you can get away with maybe monthly data or daily or perhaps hourly. And so we added a question in our discussion there about the fact that it would be really nice to have diel information, but that's not always possible. And so you have to be aware of that too. And then pick your algorithm too. So you can use some really convoluted, amazing algorithms that might be very robust for what you're looking for. But for some questions, you might be able to get away with a chlorophyll A algorithm. So don't get tangled in the weeds, in the details. So just to wrap things up, just a reminder that remote sensing is awesome. It can provide time series and large scale estimates for some things, but there's also technical limitations. And so you have to be aware of those. And then your background knowledge is key, your phenology, your information about when they might or might not bloom so that we can see that from space. You need to be aware of which satellite mission fits your particular needs and your question. And remember that we have to have something to measure from space. It's not witchcraft. We need some sort of signal. So think about all the different approaches that we discuss and what might be a good way to go. And then something that I'm trying to work on, I'm trying to start a hyperspectral library that can be shared, because when you try to start doing some experiments, one thing you found is that there's not a good library of references that people can look at. And with that, I'll thank you all for listening, and I'll take any questions, and thank you for the opportunity and having me here today.
Excellent. Thank you so much, Sarah. Congratulations on your recent grant and good luck studying Noctiluca. And I definitely love your library that you're trying to create of all of the hyperspectrals. Don't even have time to ask my own question. Looks like I'll turn it over to Becky real quick.
Hi, thank you very much for a great talk, Sarah. I don't really have a question. I just wanted to let you know that if you're interested in, well, you are interested in Noctiluca cultures, you should contact Helga Gomes at Mont Doherty.
Yep.
I believe she has Arabian Sea isolates. I don't know if they're still going.
Okay, thank you. Yeah, I know they've done a lot of work. She and Joaquin goes, but I don't know if they have them. Like I said, I'm just starting, so thank you for the tip.
Are there any questions? Any other questions? Because if not, we'll move into our breakout groups. I'll actually post here. We've got some discussion questions that I added some of my own, but Sarah and Jessie came up with some great discussion questions for everyone. And I've created three breakout groups randomly assigned. Holly, Becky, and myself will be the leaders in those groups. And then the first page of this document has the questions, and then the subsequent pages has space for each group to type out their ideas. We'll go into these breakout groups for 20 to 25 minutes, and then we'll come back together around 12:15 and have a little bit of a report out from each group to see what everyone discussed. So I will I'll just open up these breakout groups right now and see everyone back here.
Hey, Nicole, are you with us? Hopefully your computer is being...
Yes, I am. I'm here. Yes, it took me a while to get back from the breakout group. All right, I hope everyone had as fruitful of a discussion as our group did. So I'll just jump right into it. If someone from group 1 has ever decided to be the reporter, would like to just give us a quick overview of what you guys talked about, any specific highlights from your discussion.
Great. I'm Ashley Maloney, and I volunteered to do this, but Holly and Jessie will definitely jump in if I forget something. So we started off trying to remind ourselves why we're interested in satellites and mixotrophy, and I was just saying that, listen, the big goal of any ocean research is to be able to look at the full spatial coverage and know everything you want to know all at once. And then the other side of that, a long-term idealized goal would be to also be able to look at everything through time in the past and the future. So obviously we're not going to get all of that all together, but that was the goal of trying to brainstorm how we could get satellites involved. So our discussion went to interesting phytoplankton in Antarctica that seemed to be underestimated by satellites. And why is that? Maybe it's because of some sunscreens and protective chemical layers that they have, or maybe it's because of their frustrals of their diatoms. So we started talking about lots of issues and problems, and we ended up looking at a table of different types of pigments that are in different types of phytoplankton to see how well we could suss that out. But then we kept falling back onto the issue of like, how are you going to be able to use a satellite to see a mixotroph at all? So then we started designing an experiment. So our experiment would be something like grow a mixotroph, but either have it be a total photoautotrope, so don't feed it any prey, or be able to quantitate like how mixotrophic it is. Maybe at 50% of the predators are engaging and eating and have eaten something, and then shine a radiometer on that either in the lab or on a pond with a drone, or a big pond with an actual satellite, and see if there is any signature at all. Like maybe this doesn't exist, but my idea physiologically was thinking, well, if a cell is eating bacteria and it has bacteria in it and it's digesting bacteria and it's getting a little bit bigger maybe, and different enzymes are working, maybe that just changes the signal that you would pick up with a satellite. So that was our experiment that we brainstormed at the end. Did I miss anything, guys? So I'll pass it on to group 2.
All right, that sounds great. The reporter up from group 2.
Okay, so I took notes on this, and yeah, we had a lot of discussion on the different aspects of remote sensing and what it could tell us about the cells that are present. It comes down in the summary to you need to have good environmental predictors, or you need to have good optical signals, and they need to occur over a region that is a proper size. So really small scale things are difficult still at this point. They will get better. Mixtures are really difficult to sort out. So right now, unless you have a signal or something that points to your mixotroph, it's going to be a little bit tougher. And so designing those experiments is a very fun idea. So see what we can do there. The strengths really are that we can look at large scales in regions, and we can get long-term data as well as relatively frequent sampling. And finally, what was one of the other things that came up here? Right, we talked quite a bit about this, the whole aspect of environmental signals. So using things as proxies rather than actually just measuring mixotrophs or trying to measure mixotrophs. Knowing either the environmental conditions under which they become abundant and active, or if there are other taxa that are present that could serve as a signal that your mixotrophs are there. Those are things that could be pursued in, as opposed to trying to detect them specifically. So if I missed anything, please, group 2, jump in.
We also talked a little bit about the DL patterns and yeah, and whether we're seeing changes in our signal because they are being eaten, degraded, gone down in the water column. That can become a little tricky too. And it seems that we need to figure that out in situ before we really do a lot of work with remote sensing too, or we can combine the two.
Yeah, it came down to, we still need to know more based upon microscopy and lab experiments and field measurements before we can really start applying. So again, it's what are your questions? And then what's the most appropriate thing to measure them with?
And Sarah gave us 2 resources for if we want to learn more about doing more remote sensing. She told us about 2 great courses that we could think about sending a grad student potentially to, that was going to dig in deeper than any of us might. One was at Cornell and one was in France, and they were both sponsored by OCB. She can tell you more details, but I thought that was really helpful to know about.
Yep.
All right, great. Thank you. So for the last group, I nominated Karen at the last second to be our reporter.
She literally nominated me when there were 2 seconds left on the clock, and all I had time to say was, okay. Oh, Nicole really knows how to make people say yes. So yeah, sorry I was late to these talks. I missed the talks, but I will look at them online. So thank you for well, I should say, please forgive me for being late. So our group, I think I would echo just first of all a lot of things from the previous group where we talked about the limitations of satellites. Even just estimating things like biomass and chlorophyll A biomass in the satellite data is, even on a large scale, has a lot of uncertainty. And so we talked about how if you zoom into smaller and smaller scales, that uncertainty gets larger and larger. Of course, the hyperspectral analyses, it seems like, are a good place to start. That as has been published, sometimes we can use that to look at large mixotrophs in places where there's a large bloom of them. So it's a really clear signal. But of course, you need the in-water data to validate that. I had thrown out there that we could use a full suite of satellite environmental measurements if we could have really good correlative models for mixotrophs in the water as it relates to water column structure and things we can actually measure from satellites like sea surface temperature, even, and I think sea surface height could be used to get the physics of the water column. So I think there are things we could use to understand the conditions that would give rise to a large proportion of mixotrophs in the community. But of course, we need first, that's like way down the line. We would first really need all of the in-water measurements to get at that and have any sort of accurate estimates from a correlative type model. So we all kind of got on our soapboxes for a second and said, we need more data. Let's see. And of course, it goes back to the issue of we can maybe estimate the likelihood of mixotrophs being there. But again, it's such a high resolution temporal measurement to know whether things are actually doing mixotrophy. So again, mixotrophs versus mixotrophy. And I think we kind of, well, we'll probably never get to that from satellite data, but we don't want to give up on satellites. We think they're useful tools. That was our kind of our closing remark. We think that as new sensors come out, there's things like LiDAR and things going into the future as we start to generate new information from satellites, which they're doing all the time. We can probably use them to study mixotrophs, but right now the focus should really be on in situ data, experiments, things like that. Does that summarize our? Yeah. Okay.
I think you landed on the right takeaway too from our discussion.
And I want to add to that about new satellite missions. CubeSats present with the right platform to do these kind of experimental missions. We can actually increase the spatial resolution relatively easily, but it comes with other trade-offs too. But just think about it that way. They're low cost, and we can, if you give us enough information of what to look for in our signal, we can potentially come up with a new satellite mission.
All right. Jessie and Sarah, Ashley put one last question in the chat, kind of off of like what we were talking about. If you had to pick one satellite as a representative for phytoplankton in a figure, you mean the satellite data, Ashley? Yeah. Which one would you pick?
No, I mean like the actual satellite.
Oh, the best satellite
In the
okay.
sky. Yeah.
The satellite in the sky is representative for phytoplankton. Which one's the best?
I would go for OLCI, OLCI Sentinel-3. There's two of them, 3A and 3B, and it's by the European Space Agency. I'm just going to throw out, there you go. There's my vote. What do you think?
Well, I'm more interested in hyperspectral ones, but yeah, OLCI has really opened up a whole line of work in terms of spatial resolution and the bands that they picked up For example, people doing water quality in freshwater or very coastal regions, you need those bands in the red that we didn't have before. But yeah, I'm more excited about Pace and Glimmer just because they're in my background too, my backyard, and hyperspectral. But one of these days I'm gonna get a hyperspectral CubeSat. Well, there's one actually. I haven't looked into the data. Oh, I'm spelling Glimmer for you. And the PI is Joe Salisbury from New Hampshire.
Yeah, I'm also excited for Pace with that hyperspectrals. All right, well, we are at 12:30. And so I would just again like to thank Jessie and Sara for their time and their knowledge that they brought to the group. Very, very interesting discussions. I learned a lot. I thought we had a great discussion. I hope everyone else felt the same way. Keep posted for our next meeting in September, after we get to meet as a group and settle on what we're going to talk about next. So yes, everyone enjoy the rest of July and your summer. And I hope you're in a place where you're staying cool, at least, because it's hot.
Thank you. It was a pleasure.
Bye, everyone.